Hi, my name is Shen. I'm a computer science PhD candidate at Washington University in St. Louis. So I'm gonna talk about this joint work with my advisor, Gary Stormo. So this is about deep unrolling for regulatory genomics problems. So first, the summary. Our method is interpretable and it is fast, and it can be used for downstream regulatory genomics tags such as sequence classifications. And we're gonna show some of the result on motif discovery using the approach that we have. So let's go to the formulation of the problem. We're gonna assume that encoded DNA streams can be approximated as a sum of linear convolutions shown in equation 1. Here, D is a filter and X is a sparse code. We can turn this into a problem called convolutional dictionary learning shown in equation 2. And in equation 2, you can see that the first term here is the data feeding term, and the second term here is the regularization term that induces a sparsity on the code. And here in the data feeding term, D is the concatenated version of all the filters together, and X is the concatenated version as well for all of the sparse code. And I simply indicated the constraints as CD and CX. And this idea, convolutional dictionary learning, is in the following figure. As you can see, the superposition of the filters approximates the set of DNA sequences. So there are many ways that we can carry out the optimization. The typical way to do this for this convolutional dictionary learning problem is through alternating minimization. So this problem can be formulated in two convex subproblems. One is called sparse coding and another one is called dictionary update. In sparse coding, we simply fix D and optimize X. And in dictionary update, we fix X and optimize the dictionary D. And we're simply gonna use gradient descent to do the update. And one of the idea following Gregor and LeCun is that we can actually unroll the gradient descent to update these two subproblems into a neural network. So what we do here is that you can see that the encoder and the decoder mimics this process of alternating minimization. And in the encoder, we do the proximal gradient descent to update the sparse code X. And in the decoder, we actually perform mirror descent to update the dictionary D. So altogether, the forward propagation of this network produces an updated sparse code and an updated dictionary D at the end of the forward propagation of the network. We formulate the objective of the network as the following equation below. So the first term in this objective is the reconstruction loss. And the second term here is a coherence regularization on the filters. So this coherence regularization ensures that the filters are not too similar to each other. So let's go to the results. So it's actually pretty fast to train the network to obtain a usable representation. So we set the model with 95 filters and we train this on the GPU with 25 epochs. And you can see that the training time actually for 100,000 DNA sequences, and each of the sequence is 100 base pair long. It takes about 10 minutes to obtain a quote unquote good representation. And we can do some inference on this representation, which is from the sparse code, and then do motif finding. So some of the result we have is in this picture below. So on the left, you can see these are the motifs from the JASPAR database. And on the right, we have the discover motif from our method through inference. You can see that the discover motif matches the published one. And additionally, we find some other motifs that, for example, the long motifs in the third and in the second and the third data set actually correspond to some transposable elements. The third motif corresponds to the transposable element that is actually a published one. For more details of that, you can see citation 2. So in summary, we present a fast and interpretable deep learning approach for problems in regulatory genomics. This is based on the unrolled version of the convolutional dictionary learning problem. And our approach scales, and the experimental result shows that we find not only the published one, but also the additional motif there are correspond to transposable elements, and we also find some motifs that we can characterize variable spacing in between them. If you are interested in this, I'm happy to talk more about it. And thank you for your time, and I'll see you in the conference.